So cytoplasm is the area where mostly all the cellular activities goes on. In nucleus, there is nuclear membrane present or absent. If nuclear membrane is present, it is known as eukaryotes, and if it is absent, it is known as prokaryotes. So now we can discuss that. Now there are different shapes of cell present, like in if RBC it is biconcave, in RBC it is biconcave. For a nerve cell it is branched. Nerve cells are branched and long. Tracheids are elongated. Mesophyll cells are round and oval. Epithelial columnar cells. So there are different shapes of cells present. Ribosomes, cell organelles. Mostly ribosomes are present in both prokaryotes and eukaryotes. It is a non-membrane bound organelle where protein synthesis takes place, which helps in protein synthesis. Now we can discuss about RH Whittaker 5 kingdom classification. So RH Whittaker 5 kingdom classification, that is Monera, Protista, Fungi, Plantae and Animalia. These are all prokaryotes. Fungi can be Monera is a prokaryote. Monera is a prokaryote, and these are all eukaryotes. It is unicellular. Monera, Protista and fungi are unicellular. Fungi can be multicellular as well, mostly multicellular. In Monera, cell wall is present. In Monera, cell wall is present. In Protista, cell wall is present in some and absent in some. In fungi, cell wall is present. Plantae, cell wall is present. In Animalia, it is absent. Monera is mostly phototrophic. It is mostly phototrophic, that it makes food with sunlight. Heterotrophic and autotrophic. Protista is mostly heterotrophic and phototrophic. Fungi is mostly heterotrophic. Plantae is phototrophic. Animalia is heterotrophic, that it depends on others for the food. Mode of nutrition for Monera. Is mostly that it absorptive. In Protista, it is absorptive and ingestive. In fungi, it is absorptive. In Plantae, it is mostly absorptive. In Animalia, it is mostly ingestive. Motility. In motility, it can be Monera can be motile or non-motile. Same goes for Protista, motile or non-motile. Fungi are mostly non-motile. Plantae non-motile and Animalia are motile. In a prokaryotic cell, examples of prokaryotic cells are bacteria, blue-green algae, mycoplasma, and PPLO. So, what does PPLO stands for? Any guesses? So, okay, I'll only tell. It is pleuropneumonia like organism. Pleuropneumonia like organism. Pleuropneumonia like organism and mycoplasma. These are few of the examples for prokaryotes. In prokaryotes, there is a cell wall which is made up of peptidoglycan. There is a cell membrane and a genetic material which is naked. That is no nuclear membrane. What are the types of genetic material? It is genomic DNA or plasmids. There are two types of genetic material in prokaryote, that is genomic DNA and plasmids. What does plasmids do? It gives a unique phenotypic characteristic to the bacteria. Example, antimicrobial resistance. So cell membrane is generally consists, it is a fluid mosaic model which is mostly made up of lipid plus protein. Lipid, it is a bilayer phospholipid. Lipid is a bilayer phospholipid, and protein is, it can be integral and peripheral. It is a quasi-fluid nature. Lipids, which helps in lateral movement of proteins. Now, how this lateral movement of protein aids? It helps in cell division, endocytosis, secretions, different type of secretions, and formation of intercellular junctions. Cell growth. This quasi-fluid nature is semi-permeable. The cell wall is semi-permeable in nature, that it helps in active transport or passive transport. Active transport is against the gradient and passive transport is along the gradient. In eukaryotes, the nucleus does have a nuclear envelope and the genetic material is inside it. So moving on to cell wall. Now cell wall Where is it present? It is mostly present in fungi, plants, and not animals. Animals में cell wall नहीं होता. It is a non-living rigid structure which is mostly made up of peptidoglycan, but in different organism it is different. Like algae में cell wall का composition is cellulose, galactins and mannins, and in plants it is cellulose, hemicellulose, pectins and proteins. There can be two types of cell wall, primary or secondary. What are the functions, different functions of cell wall? First of all, it gives shape, it gives protection from mechanical damage or infection, it acts as a barrier to undesirable molecules, and it helps in cell to cell interaction. Next cell organelle is mitochondria. Mitochondria is the most important cell organelle which a cell has because it is the powerhouse of cell. It produces energy in form of adenosine triphosphate. It is a double-layered structure which contains an extra nuclear DNA, which is a single circular molecule, and it contains RNA molecules and ribosomes. There is cristae. Here is extra chromosomal DNA present. Moving on to the next organelle, chloroplast. Chloroplast is only present in Plantae, mostly present in Plantae, and it is responsible for photosynthesis. It is absent in animal kingdom. It contains chlorophyll. There is stroma. Stroma contains enzymes which helps in carbohydrate and protein synthesis. It also contains a small and double-stranded circular DNA. And the stroma contains small So what are the major difference between animal cell and a plant cell? Major difference between animal cell and a plant cell. Animal cell contains centrosome, which helps in cell division. It contains lysosome, which helps in lysis, and centriole. Plant cell contains chloroplast, cell wall, and plasmodesmata. Nucleus. Nucleus contains of nuclear pore, nuclear membrane, and nucleolus, chromatin. In this nuclear pores there is entry and exit of RNA and protein. This is nuclear membrane, this is nucleolus, and this is chromatin. Nucleolus is not membrane bound and it helps in rRNA, ribosomal RNA synthesis. So we have seen all the important cell organelles, its important functions. Few of the cell organelles which is remaining and their functions are endoplasmic reticulum, in which it is of two types, smooth and rough. Smooth is involved in lipid synthesis and rough is involved in protein synthesis. Golgi apparatus is related with packaging. Vacuole storage. So at the end we'll just discuss what are the main difference between eukaryotic and a prokaryotic cell. Eukaryotic cell it has a well-defined nucleus, membrane bound organelle. Example of eukaryotes are animal, plant and fungi. In prokaryotes examples are bacteria. Nucleus is not well defined with no membrane. It is mostly unicellular. Cell size of eukaryotes vary from 10 to 100 micrometer, and in prokaryotes it is mostly less than 5 micrometer. And DNA replication of eukaryotes is highly regulated. The entire genome replicates at once. Generally more than one chromosome. In prokaryotes it is one long single loop of DNA with plasmids. In eukaryotes large ribosomes, in prokaryotes small ribosome. Growth rate in eukaryotes is slow. Growth rate in prokaryotes is faster. Organelles in eukaryotes are membrane bound and present in prokaryotes.